Okay, so I'm going to explain the first database question on Z-maize on the corn. So it's said to begin with that it used two varieties. Often when you get breeds of plants or animals, the process of selectively breeding them actually creates varieties that always true breed. You always get the same offspring. And this means that their alleles are generally homozygous. So we started with the two parents, the coloured starchy breed and the white and waxy breed. And we know that 100% of the F1 offspring were coloured and starchy. So that tells us that coloured and starchy were the dominant alleles and white and waxy were the recessive. So therefore I could work out very easily that the first parents had big C, big S, and the other parent, the white and waxy one, had little c, little s, because that was 100% recessive. Because we know that, because none of the offspring were white and waxy, they were all coloured and starchy. So in an F1 offspring, you always get this diheterozygous, this big C, and big S, little s. So the next thing it said is they bred two of these F1 offspring together and produced an F2 generation. So I've done the F2 offspring here. These are my F2. And all I did was I worked out, obviously, the gametes as you normally would. And then you can see that I've completed that Punnett square and worked out the ratios. And these are the normal ratios we would expect. We would expect a 9-3-3-1 ratio for dominant with dominant, dominant with recessive, recessive with dominant, and recessive with recessive. Now, the question asked, how do we know, or what can we say about the occurrence of these two particular genes? Well, when we had the actual frequencies, we got these numbers here, these actual frequencies. Do they fit this ratio? That's the question. And clearly the answer is no, because if you're to add up the total offspring together, you get 2736. If we divide that by the number of different phenotype categories, so here we got 16, and we end up with 171. So we would have expected the numbers 171, 513, because that's 171 times 3, 513 here, and 1539 here. And we can see very clearly that they do not. They're very, very different, especially these ones. There's far fewer of these than we would expect. And that's because these ones here can only be produced by crossing over. So in fact, instead of the genotype looking like this, the genotype was actually this. C and S were linked, and little c and little s were linked. And the only way we could have got these gametes being produced is if crossing over occurred, and you got recombinations occurring in that way. So that's the only way it could have happened. Okay, so that's the answer to that question.